Hi. Thanks for joining us. I'm glad you were able to attend our garden speaker series. My name is Marina Montez Ellis, the garden program specialist with the Museum of the Grand Prairie and the Champaign County Preserves District. For those of you not familiar with the area, we are located in Mahomet, Illinois, just a few miles northwest of Champaign-Urbana, home of the University of Illinois. Tonight, we begin our garden speaker series, and soon we'll hear from tonight's speaker, Megan Romberg, with her presentation, One of the Fungal Final Frontiers, Filling in the Gaps on Our Knowledge of North American Microfungi and How You Can Help. I want to go over a few items before we get started. So first of all, let us know where you're watching from, and you can tell us in the comments. We have a few people from Champaign and Charleston. So we are the Champaign County Preserves District. We are located in East Central Illinois with 7 nature preserves that covers over 4,000 acres in Champaign County. In fulfillment of our district's mission to protect Champaign County's natural and cultural resources and inspire people to care for, enjoy, and explore the natural world, the Champaign County Forest Preserve District recognizes its responsibility to acknowledge the indigenous people who occupied the area long before there was a need for these county preserves. We currently work to preserve and tell the story of the lands of the Peoria, Kaskaskia, Wea, Miami, Kickapoo, Michigami, Pinkashaw, Muskokan, Sauk, Meskwaki, and the Potawatomi nations. Native Americans shaped the landscape that the forest preserve sits within. We must recognize that the forest occupies the ancestral, traditional, and contemporary lands of the people previously mentioned, and that they were the first stewards of the land. And for that, we honor and respect their place in the history of Illinois. The Museum of the Grand Prairie is now open for the spring, where we will be open every day from 1 to 5. And as always, admission is free. So this is the first of our garden speaker series for 2023. We do have another presentation coming up in April on the 16th. It's a Sunday. It's called Recycling in Your Community, an overview of the Landscape Recycling Center. We'll have Derek Terry come and talk to us at the museum. It's an in-person presentation. And if you want to get more information, just look on our website or our Facebook pages. Also, our summer camps are now open for registration. So you can go to our website at ccfpd.org for more information and links to register. And as always, for updates and more info on all our programs and what's happening throughout the museum, find us on social media or visit museumofthegrandprairie.org. Should you have any comments tonight, be sure to write them in the comment section. And we want to, hopefully any questions are welcome, and we'll try to answer a handful of questions at the end of the program. So with much excitement, I'm sure you can hear that in my voice, I want to introduce a longtime friend of mine. We met in 1996 as wide-eyed 20-somethings in Miami, waiting to embark on a 9-hour flight to Bolivia, South America as Peace Corps volunteers. Today, Megan Romberg is currently a mycologist with the USDA and focuses on the identification of microfungi. She's interested in all things fungal, including documenting mushroom species diversity through DNA sequencing with the Mycological Association in Washington, D.C., dyeing wool with fungi and lichens, and designing textiles with fungal motifs. So without further delay, I'll let Megan lead us into the world of microfungi. Hi, Megan.
Hi.
So I'm going to go ahead and leave it to you.
All right. Thanks, Marina. Thanks for the introduction, and I will start presenting my screen. All right, Marina, do you see my slide?
Yes.
Yes?
Okay, you all set?
I can't see you, so I have to hear you.
Okay.
Good to go.
Good to go. We're going.
All good?
Yes. Can you not hear me? Do you see it on the screen? Do you think she can hear me? She might have muted me.
I'm not sure.
All good, Marina? Okay. All right. It's a little different from what I'm used to. All right. So I'm excited to talk to everybody about sort of my favorite subject. This presentation has kind of morphed a little bit since I started it, but I'm really just going to be talking about fungi around us and some fungi that you may never have seen. A lot of people haven't, and hopefully inspire you to get out and look for them yourself. So, and you can put this in the comments. I won't see them until after the presentation, but I am curious when I say fungus, what comes to mind? When I talk to people and I tell them that I'm a mycologist, usually I get a blank look and people say, you know, what on earth is a mycologist? And I'll say that I study fungi. And usually people have some sort of thought about what they imagine. Often people think of button mushrooms or other mushrooms that they might eat. Every once in a while, if I say I work on fungi, I have someone sort of cringe and they must be thinking of things like athlete's foot or toenail fungus, which is not what I have anything to do with. And more recently, we've been seeing a lot of fungi in pop culture. And maybe you watched The Last of Us. I did not, and saw somebody infected with cordyceps, which cannot happen, but is kind of cool to think about. Or maybe you think about yeasts and all of the good things that they do for us, including giving us wine and beer and bread and cheese. So kingdom fungi is widespread. It is an entire kingdom and has a lot of different members. And one of the things I'm going to do tonight is maybe show you some that you've never thought of. So these are some fungi through the microscope, and this is just a fun slide showing that sometimes the fungus loves you back. And that little smiley face really is a fungus. Those are fruiting bodies of a fungus that occurs on leaves of cinnamomum. And these were leaves that were coming from India into my lab. So fungi are one of the living organisms on Earth. They are in domain Eukarya because they have a nucleus. And you can see on this pedigree chart that you have fungi over here, most closely related to animals. For a long, long time, people thought of fungi as more closely related to plants, and they were studied in botany departments. But DNA sequencing, in addition to some other evidence, has really placed them as our closest relatives. So when we study fungi, it's something that we're really looking at our cousins. And it's also one of the reasons that people have a hard time controlling fungal diseases, because things that kill fungi can also kill us. So when you see charts like this, often the fungus is represented by a mushroom. And I am not going to be talking about mushrooms tonight. A lot of people do think of mushrooms when you think of fungi and use the terms interchangeably. But again, it's a kingdom and it's full of a lot of diversity. These are a few edible mushrooms, but again, we're not going to talk about these tonight. We're going to talk about what else is in fungi. So this image comes from a group in the United Kingdom, the Kew Institute, and they put out in 2018 a document called State of the World's Fungi. And it was the first really overarching document talking about what we know about fungi in the world. And I kind of liked their little chart with something that looks like a fungus. And this just represents the different groups that are in kingdom fungi. So these are different phyla. Within a kingdom, you have phyla, single phylum, and those are sort of overarching groups. And within kingdom fungi, you have the Ascomycota, which is represented by about 90,000 species currently. You have the Basidiomycota, which is about 50,000 species. And then you have some of these other phyla. The Basidiomycota contains the fungi that we commonly see as mushrooms when you're walking through the woods in the grocery store. Jelly fungi, brackets, the little shelves that you see on trees, in addition to some phytoparasites, which are rusts and smuts. Over here in the Ascomycota, you have a lot of fungi that people don't normally think about or see because they're microscopic. So you have endophytes within trees and other plants. You have parasitic microfungi, lichens, molds, which is an artificial classification, but it's just small fungi that are growing on things and rotting them. So lichens are an ascomycete fungus and a cyanobacterium or an alga. You also have this group, the mucoromycota, which are the pin molds that you might see on bread or other fruit. The zoopagomycota, which are on insects. Chytridiomycota, which are most famously known for amphibian dieback. Smuts, probably the most well-known of which is corn smut, which is eaten in Mexico and Central America, known as huitlacoche. And then rusts, which we're going to talk about a little bit more. So fungi are much, much more than the single emoji that represents them in anything with emojis, not just red spotted mushrooms. Of course, people saw mushrooms first because they are bigger than microscopic fungi. And being able to study microscopic fungi has followed the technology and the ability to look through a microscope and see these very, very tiny things. One of the first books that came out on microscopic fungi came out in the late 1800s by a man named M.C. Cook. And they didn't have cameras that were able to take pictures. So the images of the different fungi that are in that book were painstakingly drawn and painted with watercolor paintings. So you've got some pretty beautiful illustrations of what people were able to see. And it's been amazing to see similar things through my microscope. And they were actually able to see quite a bit. I currently work with a setup like this. This is my microscope, my dissecting microscope. And that's allowed me to see a lot of things. All of the images I'm going to show were taken on this setup. But microscopes have become cheaper and more available. And it's really something that people have the opportunity to use more now and get into seeing these fungi that might have seemed a little bit daunting or out of reach previously. So before I show some of these pictures, I wanted to talk about the size of what I'm talking about with microscopic fungi. This is a penicillium on an orange. You may have seen this. Under the dissecting microscope, this sort of looks like ice crystals, but it's really, it's supposed to look like a little paintbrush. That's what penicillium means. And when you look under the compound microscope, this is what you see. You see this structure here. It's called a conidiophore. These spores are called conidia, and it is very, very small. This bar here is 100 micrometers, 100 microns, and 1 micron is 1/1000th of a millimeter. So you have this really intricate structure that is very, very small. So this, of course, is a centimeter from 8 to 9. One of these divisions is a millimeter. If you imagine slicing that into 1,000 different sections, each one of those tiny little 1/1000th is a micron. So if we take the top of one of these divisions and blow it up 600 times, this is what it looks like. You start to see all the little dots, all the little colors. And again, here's my scale bar. So 100 of 1/1000th of a millimeter. So you're going to see a scale bar on a lot of the images that I'm showing. And I just wanted to sort of give you an orientation to how small these things really are. Another thing I just wanted to mention as an orientation is I will be using scientific names. Most of these fungi don't have common names. So you have to use the scientific name. And once you get into them, they're actually not as daunting. And the names are often very descriptive and really kind of boring. So one name I'll be using for rust fungus, Puccinia, was named after Tommaso Puccini, who was a gallery director and had a mycologist friend. Peronospora is a downy mildew that you might see sometimes. Peron means brooch. Spora, of course, means spore. Philisticta is a name that I will be using. Philo means leaf. Sticta means spot. And Cercospora is a name you'll see a couple of times in this presentation. And circ means rod and spora means spore. So again, mycologists aren't actually all of that imaginative when they name things. Really, they're just using the Latin to put names on what they're seeing. If you do get into this sort of thing, a dictionary like this is really useful. This one is a great one. It's got all sorts of terms and can really help you make sense of scientific names. All right, so it's mid-March and spring is on its way, and hopefully pretty soon we're all going to be seeing yards like this where flowers are blooming and the leaves are green. And at the beginning of the season, everything looks sort of green and lush and healthy. But then as time goes on, you may start to see spots appear on a lot of your leaves. And they all kind of look the same in a lot of ways. They're just spots. I've had people ask, are they viral? Are they bacterial? Are they fungal? You can't really necessarily tell. You do have some clues. Fungal diseases tend to be more circular. But you don't necessarily know until you start looking under the microscope. And the thing is, what you see here, these leaf spots that just look like some sort of illness on the leaves, once you do look at them closely, are pretty amazing. So this is a strawberry leaf. And if you were to take off a little bit of white fuzz that's in each of these spots, what you would see is this fungus, which is called Ramularia. And it has these little structures. They're called conidiophores. And these little sort of dots here are actually circles. They kind of look like donuts. And the spores are blown out of those like a balloon. And so these are the spores of the Ramularia that get blown out of these little sites and then can land in water droplets and be spread by wind to other leaves. Many of you have probably seen this on anything that you might grow in the beet family. Even if you get a spring mix from the grocery store, you might see these little circles with red around them. If you pull off some of this fuzz, what you'll see is this fungus, Cercospora, again, rod, spore. You have these little structures that have, again, these little donut holes. They don't all do that, but these two do. And they blow out this long balloon of a spore that has some crosswalls in it. And again, here's our 100 microns. So this spore is about 100 microns long. It also travels through wind, lands on new hosts. And this last one is kind of the most exciting one. This is Photinia, and it has a leaf spot on it. And if you were to take some of the structures from that leaf spot and put it under the microscope, what you would see is this. And these are the spores of a fungus that has had its name changed, but it was called Entomosporium, entomma for insect and sporium, because the spores look like little bugs. Again, they're very, very small, about 50 microns in length, but there is this incredible diversity in shape going on all around us. If you had a sunflower and you looked at the fuzz on the sunflower, or at these little orange spikes on a serviceberry, you would see this on the sunflower and these angular cells on the edge of those spikes and these little spores inside. This is a downy mildew, plasmopora, and a rust gymnosporangium. So once you start looking and you're looking at these microscopic fungi, there really is an amazing diversity. All of these are fungi that have come through my lab that I've taken pictures of. And there is just an endless array of shapes and colors and forms that you can see. So back to our guide to the fungi. Again, there are two main groups, the Ascomycota and the Basidiomycota, and then these smaller groups. So the Ascomycota are called Ascomycota because they form their spores in something called an ascus, which is a sac that looks like this. So you've got the sac and the spores inside. And an ascus is actually a bag that contains wine. So they looked at these spore-containing structures and thought that looks like a wine bag. So we're going to call it an ascus. They don't all look like that. Some are longer and thinner, but the name has stuck. And so those are called asci, and it's the ascomycota because the spores are born in those. For the basidiomycota, if you took the edge of the gill of a mushroom and looked at it under a microscope, you would see these structures with little pointy bits coming off of them. They're called sterigmata, and the spores are born on top of these pointy bits. And this is called a basidium, which means pedestal. So the names of the phyla, the names of these groups are based on where the spores are born. Just like in plants, we look at flowers. We look at the reproductive structures in fungi to give them their names. So I was going to go through a few of the microascomycetes that you might see around you, just to give you a taste and a flavor. So this is an oak tree that's outside of my lab. And towards the end of the summer, I can gather oak leaves and you'll see little spots on them. And we're not going to talk about those now, but you'll also see this sort of white fuzz along the veins. And if you were to look at that under the hand lens, you might see little black spots. Maybe you'd think it was dirt or maybe insect frass. And if you can focus in a little more closely, you'll see these dark balls that are there amongst the white powder. This is a powdery mildew because of this powdery sort of growth. But the exciting thing happens when you take these and you put them under the microscope. Because once you mount a couple of those, then you see these fruiting bodies with the asci inside and the spores. And this particular powdery mildew and some of the ones related to it have appendages all around the fruiting body that produce these intricate spirals at the end. And I don't know, I feel like it's endlessly fascinating. I never get tired of seeing these. It's always amazing to take something that looks like it might just be bits of dirt and put it under the microscope and see these really intricate structures. Here's one of them close up. And again, these things are, this whole thing is probably about 20 microns. So you could walk by this leaf and have no idea that down below you have this going on. So I mentioned the ascomycetes are called ascomycete because they produce spores inside asci. Well, they produce sexual spores inside asci. At some point during this life cycle, meiosis happens, and that's why they're sexual spores. And so this is a part of the life cycle where it's producing spores sexually. A lot of ascomycetes also produce spores just by mitosis, just by copying the cells, and that's asexual reproduction. So in powdery mildew, this is actually the form that you see more often. You see this structure conidiophore coming off, all of these conidia, these spores being produced in chains. And that's what you see actually more often than you see the really exciting sexual state. Anyone who's gardened has probably seen this in their garden. If you grow cucurbits, if you grow almost anything else, you'll see powdery growth on the leaves. And when you look at that under the microscope, you'll see these conidiophores with the conidia. To me, they kind of look like blown glass. I threatened once to make this our Christmas card because I thought it was so pretty. And when you look closely, you can see the spores coming off. They're maybe not the most exciting and intricate, but they're pretty cool nonetheless. One of the other things that's cool about powdery mildews when you start looking closely is there's a fungus that grows on powdery mildew. So there's a fungus that eats the fungus. It's called a hyperparasite when that happens. And this one has a really cool name. It's called Ampelomyces quisqualis. Ampel means grapevine, quis means what, and quale means what kind. So the mycologists that named it were looking at it and just thinking, what kind of grapevine is this? Which I think is kind of cool. When you pull those off, you see a little structure that has a stalk, and it breaks open and releases all sorts of spores that spread and infect more of the powdery mildew. Another ascomycete you might see walking around is on maple leaves. You'll see these black spots, especially in the spring. If you look at that under the hand lens or a dissecting microscope, you'll see all these little black dots. Don't seem super exciting, but once you get them under the microscope, you see the structure with a little hole. It's called an ostiole. That's where the spores come out. The spores are these chunky little things, and when you look at them closely, they have a little sheath around them and a little tail. And it probably helps them in water droplets and moving around. It's not a flagellum. It doesn't move. It doesn't flip. But they all have this little sheath and this little tail. Another ascomycete you might see is apple scab, Venturia inequalis. This is on a crab apple near me. If you look at the leaf, you see these black spots. Again, symptoms can often look very, very similar. You just see a leaf spot, you see a black spot. So if you see people posting pictures on social media or one of the sites I'm going to talk about in a bit, and they say this is this fungus, sometimes you can do that, but most of the time you really need the microscope to be able to say this is what I'm looking at. So I would take some of those posts with a grain of salt. If someone posts this and says, oh, this is definitely apple scab only based on this image, I would question that and want the microscope images of what's going on beneath. So if you make a thin section through that leaf and look at it, you'll see the conidiaphore again, that same structure I was talking about. In this case, what happens is it grows and it produces this spore, a conidium, that breaks off. It grows a little bit more, produces another one, and that breaks off. And you get this succession of scars that produce these little rings, and you can kind of follow like the rings on a tree. You can kind of see how many times this has produced spores over the spring or the summer. So that was a quick walk through some of the ascomycetes you might see around you. And now I'm going to talk about some of the microbasidiomycetes. Again, mushrooms are in the Basidiomycota, and those are big and rather easy to see when you're out in the woods or walking around. The microbasidiomycetes are smaller, but still kind of more obvious than the ascomycetes, and we're going to focus on the rusts. One thing about rusts is they have this crazy life cycle. So whereas mushrooms produce one kind of spore, when you're trying to identify a mushroom, often you'll make a spore print and you can look at those spores. Mushrooms make one kind of spore. Ascomycetes make two kinds of spores, the sexual state and the asexual state. Sometimes they make three. Rust fungi make 5 different kinds of spores, and they often have 2 different hosts. So in this case, on the right side, you have the rust fungus making this kind of spore called a spermatium. That grows and becomes something called an ascidium with these acidiospores. That goes to a different host plant, forms these structures called uridinea, and makes uridinea spores. Later on in the season, it produces a different kind of spore, a telium, with teliospores. That does what we call germinate. It produces these structures called basidia, which is why it's in the Basidiomycota. Makes these little spores that are called basidiospores that go back to the first host and start the cycle all over again. So rusts have evolved this really crazy way of reproducing themselves, finding 2 different hosts and all these different spores. But they do make for some of the most charismatic and photogenic kinds of fungi that you might see. So the first one that I was going to talk about is on penstemon or beard tongue. So it's a beard tongue blue stem or penstemon andropogonus rust, Puccinia andropogonus. So this is a penstemon. This happens to just be in my garden. In the spring, toward the middle of the summer, you might see concentric rings of these little yellow spots on the leaf. That's the other host, the andropogon, the blue stem grass. If you look at those concentric rings with those little yellow spots, you see all of these little structures that kind of look like little flowers, little daisies. Some people call them fairy cups. You have yellow inside. And if you cut through one of these and make a cross section, you can see that ascidium with the walls and all of the spores coming out. So these spores that are being produced on the penstemon are going to continue growing on the penstemon, and later in the season, the teliospores will go and infect the blue stem rust. Another fungus that I found the other summer in my backyard was the mock strawberry or Duchesnay rust, Phragmidium mexicanum. So I have a whole lot of mock strawberry in my lawn. That's this small strawberry. And when you look at it, you'll see these black spots. And again, just based on symptoms, you can't really know what you're looking at. This could be various things. So you want to go closer and you want to turn over the leaf, and you can see these orange fuzzy growths. They're called sori or pustules. And when you look at those under the microscope, you'll see the Uridinea spores, one of the spore types, that will grow throughout the season. And if you're lucky, at the end of the season, you'll see the teliospores. So this is why I say that rusts are kind of photogenic and charismatic. They make these really interesting structures. Another one that I've seen in my yard is the mist flower conoclinum rust, Puccinia conoclini. And this is an example of a rust that's common. It's around. This is a common flower. It's native east of the Rocky Mountains. But this isn't one that's very widely studied because it's not on corn, it's not on soybeans, it's not on apples. It's on a plant out in the environment. And those types of fungi tend to be studied less. Plant pathology departments aren't necessarily looking at them because nobody is putting money into them. They were collected maybe 100 years ago by someone interested in seeing what was out there, and maybe haven't been collected since. So there's a lot of opportunity to see things that are rarely seen. So this one produces some yellowish spots on the leaves, some dark, but what you really need to do is look closely with the hand lens. You see these little pustules with dark spots inside them. Look at them more closely and you can see the spores. And then under the compound microscope, you see these Uridinea spores, which is one of those spore types. And then towards the end of the summer, the teliospores. And in this case, these were not dyed. They were just placed under the microscope in a mounting medium, and this is their actual color. And you can see they have little bumps on them. And again, personally, I think that these are endlessly beautiful. I could look at these for days. Another rust fungus that's common that you might see around is May apple rust. May apples appear in the spring. There's yellow spots on them and the rust underneath. These are the esia and the little esial cups. If you wait till a little bit later in the season, you'll see these angular yellow spots. If you turn those over, you'll see these little brown spots on there. And once you put those under the microscope, you'll see these crazy spiky teliospores. They're kind of unusual. This is in a different genus, but again, a lot of fun. That first rust that I showed was the cedar quince rust on serviceberry, Gymnosperangium clavipes on amelanchier. And you'll see these coming out in the spring, all the little orange spikes on the berries. These are actually esial tubes. So that stage of esia, they've got the orange spores inside. And when you mount one of those tubes, you can start to see these surrounding cells, which are angular with the little rust cells inside. And you can see that on these cells, you have all of these ridges and these structures. And identifying some of these species actually involves looking at how many ridges you have and how high they are and things like that. So the Gymnosperangium clavipes, cedar quince rust, also goes to amelanchier, to serviceberry, and the alternate host is cedar. So you might see this goo on cedar branches and twigs. You might look at it and think that just looks like goo. But when you put it under the microscope, you see the teliospores, again, two-celled in this case, and they have these long structures, these long sort of appendages that are called pedicels. And it's not absolutely certain what they do. Maybe they also help them move with the wind or through the water, but they are beautiful. So that was just a quick, very quick overview of some of the microfungi that are around you and some of the things that are going on when you walk by a plant with leaf spots, and if you're able to look at them under a microscope. And these really represent undocumented biodiversity. So in kingdom fungi in general, there have been about 100,000, 150,000 species described. And so that includes mushrooms, the microfungi, pretty much all fungi. We estimate that there may be 1,500,000 species. Estimates have gone as high as 17 million. We have no idea how many fungi are out there. So if you look at this bar that represents how many fungi we think there might be at a conservative estimate of 1,500,000, and how many we've described, there is this endless possibility for discovery over here. Currently, a lot of fungal DNA discovery or fungal discovery is going on with DNA sequencing. And there are quite a few fungi in one of the more public databases, the GenBank DNA database, but it's still only a fraction of what was described. And more things need to be recollected, revisited, and sequenced in order to have a better picture of what's out there. As an example of this sort of endless opportunity for discovery, I'm going back to the penstemon. This is, again, the penstemon in my yard, and I see these red spots on it, these blotches, year to year. So one year I took them and I pressed the leaves, and if you were ever interested in looking at fungi through the microscope on leaves in your garden, this is one of the first steps. It gives you a nice flat surface to look at. It gives you a use for phone books that nobody has a use for anymore. Or you could use things like a flower press that you might find in a thrift store. And looking at those spots under the microscope, I saw these little fruiting bodies. Again, just under the hand lens, they're not so exciting. I mounted that, and I got another one of these little round fruiting bodies with a hole for the spores. In this case, the spores are kind of long and thin. They look like other spores that we've seen, but they're born inside a fruiting body. So because of those structures, I knew that what I have is something called septoria, which is just another one of those types of fungi, and it produces spores within little round fruiting bodies and has long, thin spores. So I went through the literature, and my septoria doesn't match either of the septoria that have been described from penstemon. So there's one that was described in 1902. There's another one that was described in 1884. And I can read the descriptions, and I can see how big they said the spores were, and my spores are much, much larger. The other thing that I can do is go back to collections. So BPI is the National Fungus Collections where I work, and I can go and look at what these collections were. This one was described from Illinois, Septoria pensistimonis. We have a sample in the collections, but it may not be the original sample. So I'm talking to an audience that is associated to or got to this through a museum. And collections of fungi are incredibly important to the work that we do. So these are cabinets and cabinets of fungal specimens. I don't know how many millions of specimens we have in the National Fungus Collections right now. But if we didn't have these to go back to, we wouldn't have a recording of what someone saw in 1902 or in 1884 when they described this fungus. You have to have that physical specimen, and you have to be able to time travel and go back in time to when that person collected that sample. This is what a specimen might look like. It's on a large sheet of paper inside an envelope with some documentation, information about it. And inside you have the specimen itself, maybe a part of it that's segregated out, some slides that may have been made at the time, and some documentation about it. And without that, we have even less documentation of the biodiversity around us. MycoPortal is a website that lists many, many collections, including collections at the Illinois Natural History Museum, the collections that I work with, collections around the world. And it's a way of us looking at what have people collected and what do we know about the different fungi that people have collected. So for example, there's this fungus called Cercospora nepete. Cercospora we've seen before. It's the one that produces those long rod-shaped spores. This was described from Catmint in Illinois in 1922. Several people worked in Illinois for whatever reason and described a lot of fungi from Illinois. So this is the original specimen called the type specimen. Here's some documentation about it. And I don't think anyone really looked at this until 2002, when a scientist working on that group of fungi came along and said, this is my opinion about it. He wrote a little note about his opinion about it. There are, at least based on the MycoPortal collection, only 6 specimens documented in fungus collections of this, even though this fungus on catnip is probably really common. But people haven't thought to collect it, haven't thought to put it in collections, and there's no modern DNA-based examination. So this is one of many, many hundreds of different fungi. I think it would be really interesting to recollect and sequence and see whether it fits into our current understanding of how these fungi are related. So this is sort of the inspirational part of this short talk. This is the man that wrote that book that had the watercolor illustrations, M.C. Cook. He lived in the 1800s to the early 1900s. And he said at the beginning of that book, in these latter days, when everyone who possesses a love for the marvelous or desires a knowledge of some of the minute mysteries of nature has, or ought to have, a microscope, a want is occasionally felt which we have essayed to supply. This want consists in a guide to some systematic botanical study. And that's how he introduced his book to the microscopic fungi, which was one of the very few at the time. L.M. Underwood came along a bit later and wrote Molds, Mildreds, and Mushrooms. And he had a great quote in that book where he says, when a compound microscope becomes as much of a household necessity as a clock or a piano, when children are early taught the nature study of everyday life and become familiar with the common things in nature around them, these ideas as to what the term plant life includes will not only cease to strike us as mysterious, but our range of available information will be infinitely extended. And I really like the idea of microscopes becoming as much of a household necessity as clocks or pianos. So this is where, if you were interested, you could come in. Start looking. Look at all those spots on the leaves around you and get a microscope. I'm going to talk in a minute about some options. Familiarize yourself with at least one group of microfungi. Record your observations in something like iNaturalist, which I will show you, and save samples. You might find a new species or rediscover an old one. Like I said, looking at microfungi has followed technology, and for a long time it was sort of out of reach. But it's pretty amazing that currently for under $500, you can get a really nice microscope. This is the main one that anyone sort of interested in citizen science hobby microscopy has been getting. It's a very nice microscope, gives clear images, has software that hooks up to a laptop, and has really opened the world to a lot of people being able to look at microfungi. I have a dissecting microscope, which they can probably cost just about the same. But if all you're trying to do is look at those little black dots on the leaves and pick them off to put on a microscope slide, something like this, a magnifying glass with a light and a stand, is adequate. And you can look at your sample, pick off the little bits, put them on a slide, and then look at them under the compound microscope. There are some resources out there if you become interested in this. MC Cook's book isn't available anymore, but this book was written by two British authors. It is more focused on British fungi, but we share a lot of the same plants. And so a lot of the fungi in here are here as well. I found it for about $70 on Aid Books and eBay. It has plants on leaf litter, on wood and fungi on leaf litter, on wood and bark, on herbaceous plants, all sorts of substrates that you might look at, and lots of beautiful drawings of what you might be seeing. Another great book out there is the Microfungi and Miscellaneous Substrates. It goes over fungi on things like burnt ground, soil, dung. There's a whole lot of fungi on dung that people get very excited about. And that one is out there as well. But one of the other sort of trends and changes that I've been seeing that's really opening up the world for citizen scientists to get involved and document what's out there is iNaturalist. And if you haven't explored it, it's a fun place to look. You can mine all sorts of information from iNaturalist. For the microfungi, it's not the best because people do tend to just put in symptom images and say that it's this, that, or the other thing. But there are a lot of people out there that are starting to put in microscope images. Annie is a friend of mine from the Mycological Association. She has one of the best handles that I've ever seen. We are the champignons. And Annie is a very enthusiastic fungus collector. She's not a professional mycologist by trade, but she is incredibly knowledgeable and spends a lot of time collecting and putting her observations in iNaturalist, and it's sort of an inspiration to all of us. So I was going to show a couple of her observations. So you saw some of my images through sort of a professional microscope. This is one of Annie's observations of the cedar quince rust. She goes through and does a really good job showing the landscape picture so you can see what you're looking at, a close-up of the twig, the goo that you see on it, and then the spores underneath the microscope. And this is, she has one of those microscopes I mentioned, and this is about the level of resolution and detail that you can get. Helicoma is another really cool fungus. It's not a plant pathogen, but it grows on bark, and I think she found it on a downed log. But it produces these cute little spirally spores. And another one that she found recently was on the bark of sycamore. It's an ascomycete because it produces spores inside an ascus. These spores have little appendages on the end and are just kind of an amazing thing. There's a similar fungus that's been recorded on sycamore, but I'm not sure that anything exactly like this has been recorded. So this is an example of where this could very well be a new species that she just found walking around along in the Chesapeake watershed and collecting and being curious. There are a number of internet resources out there. I'm not a huge fan myself of social media and Facebook, but I am in all these Facebook groups for fungi. So there's fungal microscopy, Ascomycetes of North America, Ascomycetes of the world, rust mutts and other phytoparasitic fungi. And people will post pictures and say, what am I looking at? I have no idea. And it's a pretty welcoming and supportive community in those groups. The Westerdijk Institute is in the Netherlands and has a lot of publications with really good images. And a number of years ago, I started doing workshops on identification of microfungi. And the workbook that I initially put together for one of those workshops is available online at this link. And I'm sure Marina I can send her all of these links if anyone's interested. And it sort of covers the basics of getting started looking at microfungi. All right, I'm not really sure what time it is and how much time I might have gone over, but this is one last microfungus. If you're looking at ladybugs, especially the 9-spotted ladybugs, you might see these little yellow things coming off. That is an ascomycete fungus, Hesperomyces virescens. This is the entire fungus fruiting body. Inside is an ascus with ascospores, and it attaches itself to the exoskeleton of the insect. So I'll leave you with that and say thank you.
Great. Thank you. So if you have any questions, go ahead and write it in the comments, and we'll get to those. Oh, can you hear us, Megan?
I can hear you, yeah.
Oh, great. Okay, so Pat says, do you have a favorite fungi?
Every week.
Fungi every week, huh?
My favorite fungus is the most recent one I've identified. I like the ones that make me work a bit. So sometimes I'll get things into my lab and I'll have to go through a lot of different resources and look through different literature. And those are the ones I remember, the ones that make me work.
That's great. So I just have a few questions. So what was your motivation, or what motivated you to get into mycology?
So my undergraduate major was botany. And, you know, it's funny because I studied plant biochemistry. I didn't really study sort of putting names on plants and going out into the woods and anywhere trying to put names. And the names never stuck. I tried to learn the names of all the plants and they never stuck. But then I went to graduate school for plant pathology. And a lot of plant diseases are caused by fungi. And once I got into the lab and started looking at them through a microscope, it was like, I don't know, a chorus of angels opened up or something like that. From that very first time looking through the microscope, I knew that's what I wanted to do.
Oh, that's great. And so do you have any resources that, any books or websites that, you know, the audience might, you know, look into, that maybe something that inspired you to follow mycology?
Well, for me, again, it was just really looking that first time. So, but if you get on iNaturalist and start looking at people's pictures, or if you're on Facebook and you get on fungal microscopy and see what people are posting, or Ascomycetes of the world, because they posted a lot of pretty pictures on there. Some of that might inspire you to do your own. I find a lot of people sort of enter mycology through mushrooms, and they get interested in walking around and finding mushrooms and then want to start to look at them under the microscope, because there are a lot of mushrooms that you can only identify by looking at spore characters and things like that. And then once they look through the microscope, they see this whole world open up and want to explore it further.
Great. So we do have a question. Put this out there. Nat Martinez says, is the ladybug sick? And also fungi doesn't necessarily mean illness then, right?
Right. No. So this group of fungi, the Labubeniales, that occurs on the exoskeleton of insects, it doesn't seem to really sicken the insect. It may have some physiological consequences, but they fly around, they reproduce, they eat, they seem to do everything just fine. So having the fungus present doesn't necessarily mean they're sick. And a lot of plants, you'll see leaf spots over and over again every year. And the plants have co-evolved with the fungi that are on the leaves. And it doesn't seem to necessarily harm the plants, and in some cases may actually be beneficial in some ways.
Okay, I have a question. You showed that collection, you had volumes of specimens collected. Do you know of any standardized specimen that has been reintroduced into the environment for whatever reason? Do they bring them back? I guess so.
Kind of like Jurassic Park for
yeah.
No. So when something's in a collection like that, they're devitalized. So those have all been dried and really should not be growing. So in those samples, like I showed you, that are in the envelope on the paper, those have been pressed and completely dried. And actually, generally after a few weeks, most fungi will not rehydrate and be able to grow again if they've been thoroughly dehydrated. There are culture collections of living cultures that are kept under liquid nitrogen, and you take them out and you can regrow them if they are fungi that you can grow on artificial media. So there are a lot of fungi that you can grow on artificial media, but there are also a lot of fungi you can't. So rusts, for example, you can't grow on artificial media. They have to have a living host. Powdery mildews have to have a living host. And there are several other fungi that have to have a living host. So you can store some fungi, and the group in the Netherlands, for example, has fungi that they've stored for 100 years. They kind of take them out every once in a while, regrow them, get them to live again, and then put them back under liquid nitrogen. So you can store some fungi for a long, long time under certain conditions and then bring them back to life. But collections like an herbarium collection, those are usually devitalized.
For reference, I see.
Yeah.
So we have another question. Laura Jacob says, could you tell the name of the word root book you mentioned?
Sure. The author is Donald Borwar, and here I can probably get to, I could type it in the, so it's the Dictionary of Word Roots and Combining Forms. I don't know if I can type. Can I type that anywhere?
Let's see, maybe
you can write it in there.
Okay, go ahead and give me the name again.
It's the Dictionary of Word Roots and Combining Forms.
Oh, I have to type dictionary. What was that? Dictionary of
Word Roots and Combining Forms. And the author is Donald J. Borror. Borror.
Borror.
Borror. Two R's in the middle. Yeah.
Okay, I put that in the chat so you can see that. And so I thought there was another question.
Will eating a leaf infected with a microfungus make you sick?
Yes.
No, probably most likely not. So one thing you notice when you start looking at things under the microscope is there's a whole lot going on. If you look at a leaf that you think looks like just a plain green leaf and you look at it under the dissecting microscope, you'll see fungal spores that are just kind of hanging out that have landed on it from other places. You'll see little bugs crawling around. There are mites everywhere, and you've probably eaten more than your fair share and you haven't known it. So there's actually a whole lot going on things once you start looking at them under the microscope. And so you have eaten, most assuredly, you've eaten microfungi. Yeah. If you've got a leaf like the beet leaf that's covered in Cercospora, you may not want to eat that. And of course, when you have an orange that's got penicillium on it, you can kind of taste that. It's gotten sort of this rotten, fermented taste. So, if it's actively growing and covered in fungus, you probably aren't going to want to eat it anyway. But they're everywhere. You're breathing them in all the time.
Well, great. It looks like that's the end of our questions. I want to thank you, Megan, for this fascinating presentation of microfungi. So that's it for our program. And remember, you can look on our website, ccfpd.org, or our Museum of the Grand Prairie website for more information on events like this. Thank you. Good night.
All right. Good night.